Uh, you know about the human genome. We have about 46 chromosomes going to phase of chromosomes. One XY in male and one phase of XX in females. We talk about the categories of genetic disorders, single gene and chromosomal complex. Actually, what we can talk about the pattern of inheritance is about single gene disorders. For multifactorial or complex or multi-gene disorders, you cannot have a pattern of inheritance. We have actually Mendelian disorders, which are actually based on Mendel law. And by this Mendelian disorders, we can talk about actually pattern of inheritance and can calculate the risk for each condition. But we can, for common disorders or actually common variants, we cannot have risk calculation. We can just say the actually condition will be more common in one families than general population. Before we talk about the pattern of inheritance, we need to talk about the complete and incomplete penetrance or dominance. Complete dominance are phenotypes that are indistinguishable in heterozygote and homozygote states. For example, this is actually for autosomal dominant disorders. For autosomal dominant disorders, if one allele is mutated, we will have the phenotype. But in complete penetrance, based on the phenotype, we cannot say this individual is heterozygote mutated or homozygote mutated. We cannot distinguish between the homozygote and heterozygote state based on the phenotype. But in incomplete penetrance, usually the phenotype is more severe in homozygote condition than heterozygote condition. It means for autosomal dominant, if one person has two mutated alleles, the phenotype will be more severe than if he or she has just one mutated allele. One pattern of inheritance is autosomal dominant. Usually in theory, autosomal dominant inheritance is the simplest mode of inheritances, which is actually expressed in heterozygote condition, as we said. In homozygote condition, the situation is unknown because for some homozygote situation, you do not have any alive person. It means if the person is homozygote for that situation, he or she will not be alive, so we cannot talk about the phenotype in homozygote situation. But in some states, the homozygote situation is more severe than actually heterozygote. More than half of the Mendelian disorders are inherited as autosomal dominant. The incidence in some of the autosomal dominant disorders in different population is different. For example, familial hypercholesterolemia is 1 in 500, myotonic dystrophy is 1 in 550, and Huntington disease is 1 in 3,000. There are some characteristics for autosomal dominant that we are going to talk about there. One characteristic is the phenotype usually appears in every generation. It means the disease is transmitted vertically from first generation to second, from second generation to third, and the disease is transmitted vertically. Usually, phenotypically, normal individuals do not transmit the disease to the offsprings. If it means if some individual is normal or autosomal dominant, we expected that the disease will not transmitted to their offsprings. There are some exceptions for this. One is new mutation. It means the parents are normal, but new mutation happen in the gametes of the offspring and they will be affected. This is not transmitted, just happen because of the new mutation. And the second actual exception is non-penetrance. What is non-penetrance? The disorder is not expressed. It means individual has mutation, but he or she doesn't have any phenotype. Okay? But he or she can transmit the disease to the offspring.
Carrier.
Okay? Because of the what?
They're a carrier.
No, carrier is for autosomal recessive. Okay? This is not carrier state. He or she has the mutation, but he or she doesn't have the phenotype because of some actually situation that some of them are unknown. Okay, he or she has mutation, but doesn't transmit, but doesn't have the phenotype, but can transmit the mutation to his or her offsprings. Okay, the disease doesn't express in the individual because of some conditions. The second characteristic of autosomal dominant is males and females are almost equally affected. If both sexes are affected in the pedigree. When we draw the pedigree, then we can see males are affected and females also are affected, almost equally are affected. And the next characteristic is there is male-to-male transmission. It means there are some fathers in the pedigree that they transmit the disease to their sons. Okay, male-to-male transmission. You will understand about this male-to-male transmission when we talk about the X-linked transmission. Okay. And also, for each affected individual, there is a 50% chance of having an affected child. This is a characteristic autosomal dominant pedigree. We talked about 4 characteristics for autosomal dominant. Can you say these 4 characteristics? One is the disease is transmitted vertically. Okay? Each affected individual has one affected parent. Okay? For example, this affected individual.
My passport for my room.
So we will go to him.
1D.
Who is in the 1D?
Same.
For what?
This affected individual, this one.
There's a work for bank.
No, I mean for
This individual
No,
is
bank,
affected,
bank.
this individual is affected, this individual is
There's no
affected,
point.
but they have affected parents.
There's no point.
It doesn't work?
There's no point.
Okay. This one, and this one are affected, okay, and this one, and this one are affected. They have affected mother. Okay. This one and this one is affected. They have affected mother. This one is affected, and this one is affected. As affected father. These three individuals are affected. They have affected
Mother.
father. So
Yeah.
each affected individual has one affected parent. The disease is transmitted vertically. The second characteristic was both sexes are affected almost equally. Okay, we have males 1, 2, 3, 4, 5 males that are affected in this pedigree, and we have also females 1, 2, 3, 4, 5, and 6 females that are affected. Okay, both sexes are equally. And another characteristic was there is
Male
male transmission.
transmission.
Okay, in which part of the pedigree we have male
The third.
transmission?
Second and third.
Is male affected as affected child who is male, right? There is male to male transmission, right? Okay. These are characteristics of autosomal dominant pattern of inheritances. Also, the risk of actually transmission of autosomal dominant to their offspring is there is if one normal parent might be affected parent, and there is a 50% chance of having an affected child. Affected one, and 50% of having normal. If two parents are affected parent marrying affected parent, there are 50% of having affected heterozygote, okay? 25% of having affected homozygote, and 25% of having a normal child. This is an example of autosomal dominant inheritance, which is achondroplasia. This is that known as incompletely dominant. They have some skeletal disorder of short limbs, dwarfism, and large head. They have normal IQ. They have normal lives within actually their physical capabilities. If marriage happens between two achondroplastic individuals, which is common usually, this marriage, they will have 50% achondroplasia, which are heterozygote, and 25% normal, and 25% homozygote, which usually they are not alive.
Why is this called incomplete? Why is this called incomplete?
Because we cannot distinguish the genotype from the phenotype. Okay, we don't know this individual is homozygote or is heterozygote based on the phenotype. And also there is another incompletely dominant inheritance of autosomal dominant, which is familial hypercholesterolemia. Usually homozygote patients have more severe disease, and they have earlier age at onset, and they have much shorter life expectancy. There are some autosomal dominant disorders that are sex limited. It means both autosomal dominant and autosomal recessive inheritances have a ratio of 1:1. It means the number of affected females are the same as affected males. But there are some autosomal dominant inheritance which is limited to males. It means female carry the mutation, but they do not have the phenotype. The phenotype appears just in males. We call them as sex-limited autosomal dominant. One example is actually familial testotoxicosis. In this disorder, which is autosomally dominant transmitted, we have mutation in luteinizing hormone receptor. Usually affected boys have secondary sexual characteristic and they have adolescence growth at about 4 years of age. This is an example of sex-limited autosomal dominant pattern of inheritance. All affected are males because the disease just appears in males. Maybe we have a problem to distinguish these kind of disorders with X-linked disorders. Okay? In X-linked disorders also we have affected males. Okay? But the differences between this one and sex-linked or X-linked recessive is male-to-male transmission. This is autosomal dominant, then we will have male-to-male transmission. In this part of the pedigree, we have affected male, okay, and he has 2 affected boys. But for X-linked recessive, we do not have male-to-male transmission, okay? Because chromosome X is not transmitted from father to the boys, okay? So the differences between this pattern of inheritance and X-linked inheritance is male-to-male transmission. There are some factors that affect the autosomal dominant pedigrees. One is late or variable onset. The other one is lack of penetrance, variation in expression, and new mutation. What is the late or variable onset and how this factor can affect autosomal dominant pattern of inheritance? Do you know anything?
It comes at a late age. It like shows its phenotype at a age later in life.
Yeah. Maybe some disorders are late onset. Okay, it means they will have phenotype at age of 14 or 15 or even more. Okay. When you draw the pedigree, you have normal individual, okay? Because the disease is late onset. So at the time of the pedigree, when you draw the pedigree, he or she is not at the age of onset. He or she is less than onset. So you assume that this individual is normal, but he or she will have affected children. Okay. The other one is lack of penetrance. What does it mean? It means he or she has mutation, but he or she doesn't have any phenotype. Okay. The penetrance is not 100% in this pedigree, but he or she can transmit the disease to his or her offspring. Okay? You assume that this individual is normal, but he or she is normal, but he or she has mutation. But the disease is not appear in this individual, but in next generation then we will have a phenotype, okay, because of the lack of phenotype.
So, doctor, he or she has the mutation or disorder, but that not show to her. Or, so just he will send it, or
Yeah.
Yeah. Because of the penetrance. What's the penetrance?
You can give it to anyone.
The penetrance is the percentage of individuals who has the mutation and they
Phenotype.
have the phenotype at the same time.
Is that not a bit similar to recessiveness?
No, it's autosomal dominant.
But this like transfers to the. Okay.
The problem
But it's
that they
dominant.
make is the same, okay? Because in autosomal recessive, the parents are normal.
Yes.
Okay, both parents are normal, but they have affected child.
Mhm.
In this factor,
As well.
both parents, they seems to be normal.
The phenotype is normal, but the
The
genotype is.
phenotype is normal, but one of them has mutation.
Uh-huh.
But he or she doesn't have the phenotype, we assume
Uh-huh.
that both are normal. But one of them has mutation, then can transmit, so it make us to make a mistake
Mhm.
and to think that this pattern of inheritance is autosomal recessive.
Mhm.
But it's not autosomal
It's
recessive.
dominant.
It's dominant, and the disease doesn't appear in one
It's
parent
lack
because of
penetrance.
the penetrance.
Mm.
And another one is the new mutation. It means both parents are normal, it doesn't have any changes in the DNA, but they have affected child because of the new mutation. So, can you find a lack of penetrance in this part of in this pedigree? Which individual has lack of penetrance?
The one in the
What was lack of penetrance?
the second
Like
the second part.
Second part,
Yes. Has
you
lack
need to
of.
say about the generation. Generation 1, 2, 3. Okay, in this pedigree, we have 3 generations. Okay, generation 1, generation 2, generation 3. Okay, and for each generation, you numberize each individual from left to right. Okay, 1, 2, 3, 4.
The third.
Generation 1, number 1, number 2, 3, 4. Generation 2, number 1, 2, 3, 4, 5, 6, 7, 8. We can address like this.
Okay, so
So, which part of this pedigree has lack of penetrance?
number 3 is generation 3.
Generation
Generation 2.
Generation
2 in number 3.
Generation 2,
In number 3.
individual,
In
number
3.
3. 2, 3. 1, generation 2, number 1, number 2, number 3. She's normal, but she has affected girl.
Mhm.
Okay? So, this is because of the lack of penetrance.
And in
Another
number 6, generation 3.
Generation
No.
3,
Mhm.
number 6,
1,
1,
2, 3, 4, 5, 6, 7, 8,
5,
9, 10. 10th actually, I think it's 10th. I was counting on the next.
7, 8, 9, 10.
Yeah, this one.
No.
No?
Normal individual has affected child, not affected individual has
That doesn't
normal
happen.
child.
Okay.
Okay? This is affected individual who has normal child. Okay? Normal individual has affected child. Another one, generation 4,
बोला था मैंने.
Actually, this one is affected. Okay?
Its parent is.
His mother, there is non-penetrance in his mother, not in this individual. Okay, you need
So
to
it's
say
above this?
generation 1, number
इसके ऊपर हो
1,
जाएगा ना,
2,
इसके
3,
ऊपर
4.
not affected.
इसमें ये नहीं है.
Then the third one, generation 1. Third from generation 1 is also
Third of generation 1 is also non-penetrance because she transmitted to her daughter and she also transmitted, she is also non-penetrance and transmitted to her daughter. So we have non-penetrance in this
So
one
it's not.
and this one and this
and
one.
fourth one as
So
well.
it's
Yes.
generation 1 in.
Generation
Number
1,
four.
4, and
Yeah.
3.
1, 4.
1,
Because
4.
she is normal but she has two affected children. Right? Another exception was new mutation. New mutation, we have new mutation in this family. Okay, the parents are normal, but they have affected child because of new mutation. It means they do not transmit the disease to the boy. Okay. She's affected because of the new mutation. So the risk for another individual to be affected, another child to be affected, will not be increased in this situation. Okay? Because this happened because of new mutation, not because of the transmission of the disease. So the risk will be low for next children, because the parents are not do not have mutation. But in this case, parents affected have affected child. So the risk for having an affected child is 50% for next child. And from this new mutation to the next generation also we will have 50% chance of affected, having an affected. So, if disease happen because of the new mutation, the risk for next affected child will not be increased. Okay, but if mutation happen because of the actual transmission, the next of the risk of having an affected child for the next generation also will be increased. Another pattern of inheritance is autosomal recessive. Usually, majority of the cases of autosomal recessive disorder have normal parents. They parents of carrier, but they do not have any phenotype. Okay. Usually, risk prediction for this actually pattern of inheritance is simple. We just need to identify the carrier individual, then we can find out the risk of carrier for each other individual. The important point for autosomal recessive is calculation the risk of being a carrier, okay, not being affected. Okay, if you find out the carrier individual, then you can find out the chance of having an affected child. There are some rules for autosomal recessive to identify obligatory carriers. The one is the parents of an individual, of an affected individual, is obligatory carrier. It means if we have an affected individual, the parents will be obligatory carriers. Okay. In this study, these two individuals are carrier, are affected, so the parents are 100% to be
Affected.
carriers. So for each generation, for the next step, for each step farther than obligatory carrier, the risk of being carrier will be increase, will be decreased by 50%. It means for these two individuals, their parents are obligatory carrier, okay, and also their children are obligatory carrier. Then each step further, we have the risk of being carrier will be reduced by 50%. Another pattern of inheritance is X-linked inheritance. About 1100 genes are known to be located on X chromosome, and 40% of them are known to be associated with disease. For males, we have just two genotypes, normal genotype or mutated genotype, because they have just one X chromosome. So this one X chromosome may be mutated or not mutated. Okay? If mutated, then we will have the phenotype. If not mutated, then we will not have phenotype in males. But in females, we have three conditions, because they have two X chromosomes. Okay? One is homozygote, unaffected. If both X chromosomes are unaffected, are not mutated, we have homozygote, unaffected. If one X chromosome is mutated, we have heterozygote condition. And if both X chromosomes are mutated, we have homozygote affected. Okay? So for males, we have only two genotypes, normal or mutated. For females, we have three phenotypes: homozygote, mutated, homozygote normal, and heterozygote. The phenotype also in female will be a little bit different than autosomal chromosomes, because we have X inactivation in females. You know about X inactivation? Females usually has two X chromosomes, okay? But in normal, usually one X chromosome is inactivated. Okay, just one X chromosome is active and express their genes. Okay. This inactivation usually happens randomly. It means in some cells, maybe mutated X is inactivated, so the normal X will be expressed. In some other cells, maybe normal cells, normal X is actually inactivated, then the expression will be dependent on the mutated X. So the phenotype is a little bit different than autosomes because of X inactivation. Some mutated, some cells which has mutated X, maybe this mutated X will be inactivated, and then we will have a little bit different phenotypes. Also, we have dominant and recessive X-linked disorders. Usually, this dominant and recessive we can see in females. Okay, because in males we do not have. We just have one X chromosome. If this one X chromosome is mutated, we will see the phenotype. If it's not mutated, we will not see the phenotype. But in females, we have two X chromosomes. If the pattern of inheritance is X-linked dominant, then if we have mutation, we will see the phenotype. If the pattern of inheritance is X-linked recessive, we cannot see the phenotype in mutated X. But because of the X inactivation, the phenotype is a little bit different than actually autosomal. So dominance and recessiveness for X-linked disorder is not absolute in females. We say X-linked dominant or X-linked recessive, but because of the X inactivation, maybe this recessiveness or dominancy is not absolute. About 40% of X disorders may be classified as recessive because they show little or no penetrance in female heterozygote. About 30% will be considered as dominant because they are penetrant in most females, and 30% of X-linked genes can be seen as heterozygotes but cannot be classified as dominant or recessive.
Check the video.
X-linked recessive usually, typically is expressed in males, but not in females. So it means it's restricted to males. If we have a mutated allele, we can see the phenotype just in males, not in females. An example of actually X-linked recessive is hemophilia. You can see in this pedigree, all affected individuals are males. We do not have any affected females, but we have carrier females. If this individual is affected, he transmitted X chromosome to his daughter, this one and this one, but they are not affected, they are carrier. This individual transmitted to her son is affected, okay, but her daughter is carrier. Again, this individual transmitted the disease to her boy, okay, her son, then her son is affected. If affected male for X-linked recessive inheritance, if affected male married normal female, we have actually daughters who are all carrier because daughters receive X from her father. Okay, they all are carrier, but they are not affected. And the sons are all unaffected because sons do not receive the X chromosome from their father, his father. Okay, so all are unaffected. If normal male married carrier female, about 50% of daughters are normal and 50% of daughters are carrier. And if normal male married carrier female, 50% of sons are normal, 50% of sons are affected. Okay? And if affected male married with carrier female, 50% of daughters are carrier and 50% of daughters are affected. And 50% of sons are normal, 50% of sons are affected. In X-linked recessive, we will have affected female if we have consanguinity marriage. What is consanguinity marriage?
Between two close relatives. Or
Two blood relatives.
Yeah.
Okay, two blood relatives. If we have marriage between blood relatives, we call them as consanguinity. In this pedigree, we have affected female for X-linked recessive. Because her mother is carrier, she received an X chromosome from her mother and her father is affected, she also received an X chromosome from her father, both chromosome X are mutated, then she is affected. There are some characteristics for X-linked recessive inheritance. The one is the incidence of trait is higher in males than in females. Usually heterozygote females are unaffected, okay, but some of them may experience the phenotype because of X inactivation. They are heterozygote, but maybe some cells that are normal, they have a normal X chromosome, maybe they will be inactivated during the X inactivation. X inactivation is another process that happens, not related to the pattern of inheritance. Randomly, in some cells, normal X is inactivated, then this cell will not produce the protein, and maybe we have some phenotype. Usually, the gene is transmitted from an affected man to all his daughters, because affected male has one mutated X, and he transmits X chromosome to all of his daughters, and then all of them will be a carrier. Another characteristic is usually diseases are not transmitted from male to male. For autosomal dominant, we have a characteristic like male to male transmission. But for X-linked recessive, we do not have male to male transmission. Usually the disease is transmitted from father to his daughter, and then from daughter to her son. Okay? Directly, we do not have male to male transmission. And we also have some proportion or significant proportion of isolated cases because of the new mutation. We also have X-linked dominant. How can we distinguish between X-linked dominant and autosomal dominant? Based on male-to-male transmission. If we have male-to-male transmission, then we can say this is actually autosomal dominant. But if we do not have male-to-male transmission, we can say this is an X-linked dominant. Each child of an affected female has 50% chance of inheriting a trait. The expression is usually milder in females because of the X inactivation. Maybe some cells mutated X will be inactivated, and then the phenotype will be actually milder. Most X-linked dominant disorders are incompletely dominant. Affected males with normal males have no affected sound because of male-to-male transmission. Both male and female, offspring of female carriers have 50% chance. The pedigree is similar to autosomal dominant, and affected female are about twice as common as affected females. Usually affected males typically have, affected females typically have milder expression of the This is X-linked dominant, a pedigree of X-linked dominance. There are a few genetic disorders that are classified as X-linked dominant. Both sexes are affected, not almost equally, but the differences between X-linked dominant and autosomal dominant is male-to-male transmission. In this pedigree, we do not have any affected male who has affected son. Okay? He is affected, he has two affected daughters, but affected female can have both affected male and affected female. Okay? But affected male just have affected female, affected daughter. We have some conditions that are X-linked conditions like color blindness, Duchenne muscular dystrophia, hemophilia, fragile X, some kind of the mental retardation, spinal muscular atrophy, some kind of them, retinitis pigmentosa, some kind of the retinitis pigmentosa. So when we want to calculate the risk, the first thing we need to do is to draw a pedigree. We'll talk about how to draw the pedigree. After we draw a pedigree, the first thing that we need to characterize is the disease is autosomal or X-linked. Okay? If there is male-to-male transmission, we can say the disease is autosomal. Or if the diseases are limited to males, we don't have any affected female, we can say the disease is X. Okay? But if the ratio between males and female is the same, we can say the disease is autosomal. After we characterize that this pedigree is autosomal or sex-linked, is this pedigree autosomal or sex-linked? Autosomal? Why? Because both sexes are affected, almost equally. Okay? Female and male, both are affected. Right? And also there is male-to-male transmission. We have 3 affected males, 1, 2, 3. And 3 affected females, 1, 2, 3. So both sexes are almost equally affected. And also we have male-to-male transmission. This affected male has affected son. Okay, there is male-to-male transmission. So this is an autosomal disorder. Okay, not sex. Okay, and the next step is to determine the disorder is dominant or recessive. How we determine the disease is dominant or recessive?
Parents will phenotypically accept.
If one parent is affected, then we can say
Recessive.
dominant. If both parents are normal, we can say this is a recessive. Okay? So the first step is to determine the disease is autosomal or X. Okay? And the next step is to determine the disease is autosomal, dominant,
Or recessive.
or autosomal
Recessive.
Okay? This pedigree is dominant or recessive? Dominant or recessive?
The recessive because equal.
This one is now it's dominant. Now it's
This is dominant or recessive?
this one is recessive. This is dominant. This is recessive.
This
Now
one.
it's
This one is dominant,
yes, it's dominant.
right? Because affected individual have affected parents.
Yes.
Okay? This one is recessive. Because normal parents have affected children. Okay? So the first step is to determine this is autosomal or X. If both sexes are equally affected, we can say this is autosomal. If number of affected males is more or just this is limited to the males, then we can say this is X-linked. And then we must determine that this is autosomal dominant or autosomal recessive. If both parents are normal, we can say autosomal. If one of them is affected, we can say it's autosomal dominant. Any question? Actually, we have another class for you before 1 o'clock. Okay. I think
Can you
compress the classes. Clinical genetics, we study the diagnosis and treatment of genetic disorders. Population genetics, is the study of genetic in the population. Pharmacogenetics, the interaction between the gene and drugs, studying pharmacogenetics. Immunogenetics, the study of immune system, genetic of immune system, developmental genetics, normal and abnormal development, and behavior genetics is the study of the inheritance of the behavioral traits. Today we are going to talk about the structure and function of DNA, which is related to molecular genetics. As we said, our genetic information is carried out by DNA, and DNA is a double-strand helix, which actually two complementary strands next to each other. They bind by hydrogen bond between A and T and C and G. When DNA wants to be duplicated, one strand actually acts as the parental strand and the other one as complementary. And then they start the new DNA strands. This is the structure of DNA. We have five carbon sugar plus 2 phosphate plus a base. Then we have the nucleotide. We have 2 strands of DNA. As we said, G is complementary to C and A is complementary to T. Usually, when we want to talk about the sequence of DNA, we just consider one strand. Okay, actually DNA structure. When we talk about the one strand, because of the complementary strands is the other strand, we know the sequence of the complementary strand. To make it easy, we do not actually sequence both strands of DNA. We just talk about one strand of DNA, then we know the complementary strand, what will be the complementary strand. The strand that we use to sequence usually is from 5' direction to 3' direction. You know what is the 5' and 3' direction? You know?
The way the DNA is coded.
It's the right route. And there it is.
When the nucleotides actually point each other by phosphate group, okay, one nucleotide is bound to the another nucleotide. When this nucleotide binds to each other, one side of this strand has carbon number 5 free, okay, and the other direction has carbon number 3, which is free, okay. When we want to read the sequence, we usually start from 5'. It means the nucleotide that has carbon number 5 free to the 3', to the nucleotide which has carbon number 3 free. So for the other strand also, if we want to read the sequence, we start from 5' to 3'. It means for this strand, we start from here to here. We say A, C, T, G, G, C, A, T, G. If you want to read the other strand, also we start from 5'. It's C, A, T, T, G, C, C, A, G, T. This is the rule for reading the DNA sequence, and everybody knows when you read the sequence of DNA, you start from 5' to 3', then you just read one strand. The other strand will be complementary to this strand, also from 5' direction to 3' direction. So this is the standard rule for reading the DNA sequence. When the DNA wants to be duplicated, double-strand DNA is separated from each other. This is one strand, 5' to 3' direction. This is separated and acts as a template strand. Okay, and new strand will be synthesized using this template strand. And then the other strand also separated from this one and act as a template, and the new strand also will be synthesized from using these templates. And then we have two copy of DNA, and then this two copy of DNA separated to each cell. As I said, to read the DNA sequence, we start from 5' direction to 3' direction. This is the 5' direction to 3', and as I said, if you want to read the other strand, also we start from 5' direction to 3'. We also have the genetic code. Maybe you know about the genetic code. You don't need to remember this code. The only thing that we need to remember is about actually start codon and stop codon. Okay. Usually the start codon is ATG. Where can I find? ATG or AUG, usually the start codon, and the stop codon, we have three stop codons, okay? UAA, UAG, and UGA. It means when DNA is translated, during the translation step. When we when we arrive to the start codon, then DNA is translated to amino acids, okay, by start codon. And then read all DNA sequence, then arrive to the stop codon, and then translation will be stopped. Okay. The only thing you need to know is about start codon and stop codon. And the other codons you don't need to actually memorize. To the genetic is actually like atom to the chemistry. You know, all our information is carried out by the DNA, and the unit of the genetic information, gene actually is the unit of genetic information, which each gene actually translate or synthesize a protein, and this protein has a function of what? To understand the genetic, you can look at this slide. This is a chromosome. Each part of this chromosome contains a part of DNA, a piece of DNA. This piece of DNA maybe has 1, 2, 3, or many or genes included in this part of the chromosome. As we said, when we talk about a part of the gene, each gene, this part of the genome has 3 genes, gene A, gene B, gene C. Each gene actually struct or translate or express a protein. Gene A, protein A, gene B, protein B, and gene C, protein C. When we talk about the gene expression, it means how this gene actually translated to a protein. And also we talk about the protein coding gene structure. We talk about the regulatory region, which regulates the expression of the gene. And also we talk about the other part of the gene, which is exon. This gene has 3 exons, exon 1, exon 2, and exon 3. During the transcription, usually between exons we have intron. Intron is deleted, and exons are joined to each other, and then we will have coding region for each gene, which is controlled by regulatory region. Each part of the gene has actually specific characteristic. For example, regulatory region, the size of the regulatory region is about 50 to 10,000 base pair. They usually regulate the expression of the gene. This regulation could be positively regulated or negatively regulated. It means express the gene or not express the gene. Okay. Usually, regulatory region located at 5' flanking region. What does it mean? We said when we want to read the DNA sequence, we start from 5' direction to 3' direction. Usually 5' direction located at the left side of each gene. Okay? This is the 5' region of the gene, and this is the 3' region of the gene. Usually regulatory region located at 5' region of the gene, very close to the actually transcription start side. But sometimes we have regulatory region in introns. It means there is a region in intron. This region regulates the expression of the gene. Sometimes the regulatory region is located at 3' location. It means 3' location of the gene has a region, we call them as a regulatory region. This regulatory region regulates the expression of the gene. But usually, most regulatory regions are located at 5' region of the gene. We also have 5' untranslated region. What does it mean? The next region is this one. This is exon 1, exon 2, exon 3. Exon 1 has 2 parts. One is in blue and the other part is in green. Okay? Some genes have some region or some sequences that they are transcribed. It means mRNA is copied from DNA, but they are not translated. It means during the translation, this part of the gene is not translated. If this part is located on 5' location of the gene, we call them as 5' untranslated region. 5' UTR. It means this region of this part of the exon is transcribed to mRNA, but is not translated to polypeptide.
It is the cap site. Is it the cap site? It is covered, that's why it doesn't look promising.
It is what?
Cap site.
Gap side? No.
Cap site. Oh,
The capping site.
Cap side. Yeah, it's something like cap. The function of this 5' untranslation is actually one of the most important functions is translational efficiency. It's something like you have a big, for example, something a big size, okay? You want to have an exact size of this, whatever it is, okay? At the beginning, you cut it to small sizes, okay? Because finding a very small size or very exact location, it might be difficult. You make a bigger size and then select the region that you want from this smaller size. It is a very big, you divided it to, for example, 5 parts, and then start the point that you want from this part or this one. Okay? It makes your work to be more efficient. Okay? If you want to select one exact point from a big, something which is big in size, it is a little bit difficult. You may have some mistakes, but if you divided it, then you can select the exact point that you want. We have exon, and this exon has a translation start site, and then we actually make a bigger size and then select the translation start site. So it's transcribed from DNA, but is not translated to amino acid. So we call them as 5' actually untranslated region. It's something like a cap. Yeah, something like a cap to have translational efficiency. Then we have a coding sequence, which is in green, exon 1, exon 2, and exon 3. Usually we have start codon, and we have stop codon for them. The size, the average size for coding sequence in human is about 13 or T nucleotide. Average size, one gene may be smaller and the other gene may be bigger, but the average size is about 1,340 nucleotide. And we also have 3' untranslated region, the same as 5' untranslated region. we have a part in last exon, which this part of the exon is transcribed, but is not translated. Okay, this is 3' untranslated region located on 3' side of the gene. We also have exons, the functional unit of the gene or the coding sequence of the DNA are called as exons. The size of the exon vary, the number of exons actually vary from 1 exon to 178. It means some genes has just 1 exon, and some genes may have more than 100 exons. But the average number of exons per gene in human is about 9. It means average, each gene in average has 9 exons in human, and the exon size, average exon size is about 145 base pair. The other part is intron, which is located between the exons. This region is not transcribed and is not translated. Okay, what is included in the structure of the gene? The bigger gene, they have bigger introns. Okay, small genes usually have small introns. The average size of the intron in human is about 3,365 base pairs. And usually introns are bigger than exons in human. Okay? Exons are shorter and introns are larger or bigger than exons in human. What is this? Human gene, right?
The translated gene.
Translated gene. Yeah, it's a functional gene or a coding region or structure of a gene. This is casein kinase 2 gene.
Subunit beta.
I just put this slide to show you how complicated and how difficult is gene structure and gene translation and making a protein, and how complicated is our body. If you just want to read this sequence, this nucleotide or this alphabet, I can give you 10 minutes time just to read this one and to see how many mistakes you will have. Just reading this one, okay? Not, for example, selecting the exon, selecting the start codon, selecting the stop codon, from exon started from here and to here, intron started from here, and not doing this. Just reading this one, okay? During the 5 minutes, 10 minutes, you can read this one and to see how many mistakes you will have just reading this one. I just want to show you how complicated is gene structure, how complicated is translation, transcription, and protein actually structure in human. This is just one gene. Okay, we have more than 20,000 active genes in our body. Okay, in each minute, many of these genes must be produced, the protein must be transcribed, must be translated, produce a protein. In other part of body, another gene must be transcribed, translate, produce the protein. Other part of the gene, another part of the body, another gene, another gene, another gene. More than 25,000 genes we have in our body, and more than 25 20,000 or 25,000 gene must be actually translated and transcribed in our body. And this is just one part, one function in our body. Cell division, cell replication, many actually works has been done in our body. This is just one gene in our body, and this is the coding region of that gene. How many exons that this gene has?
Yes.
Does this gene have?
6.
The green part are the exons. The
6
coding region. Okay? This is exon number 1, exon number 2, 3, 4, 5, 6. So this is just one gene with 6 exons. Okay? And then we have other part for this gene. This is the start codon. Okay? A, P, G, and also we have stop codon at the end of the last exon. This is TGA. And also we have exon intron boundary. What are exon intron boundary? Is a part that exon is actually started, intron is finished, and exon is started. Okay, this is not maybe some part of this exon or intron boundary are not in coding sequence, but they are important. If we have some changes in this exon intron boundary, maybe we will have some diseases. Okay, it's not included in coding region. It's not coding any amino acid, but they are important for gene translation or transcription. Also, we have another part, we call them as intron-exon boundary. Usually, exons started with, before actually starting the exon, we have the sequence of CAG before the starting one. CAG. And after the finishing the exon and starting the intron, we have this sequence. GT, GAG, GT, GAG, GT, GAG, GT, GAG. These are important because they are important in the function of the gene. This white region are not included in the coding sequence, but they are important. When we talk about the sequence of DNA, we use that sequence, this one. Okay, we extract this DNA from the cell. Okay, and then we use PCR, polymerase chain reaction, amplify this sequence of DNA using a sequencer machine. Then we will have this kind of the sequence of DNA.
Can you record this?
Then we compare this sequence with normal sequence. Okay, if there are some changes, then we will find what nucleotide has been changed. For example, in this one, we have 2 nucleotide A and G in this part. Okay.